Hello, and welcome to the ParAqua Podcast, the science podcast that shines a light on the newest and most interesting research being conducted in the fields of aquatic and marine ecology. My name is Joe Money, I'm a PhD student and the host of this podcast. Today I'm joined by Professor Kevin Lafferty from the US Geological Survey, based at the University of California, Santa Barbara, and together we'll be discussing the role of parasites in ecosystems and how environmental disruption can impact host-parasite dynamics. Kevin, how are you doing?
I'm doing great, Joe. And thanks a lot for your interest in the research. I'm looking forward to talk to you about it.
Oh, no problem. I'm very looking forward to this episode myself. How have things been recently with your work and your research?
Well, I'll just say it's an exciting time to work in the U.S. government as a scientist. We've had a change in leadership, and so
Mhm.
we really don't know where the future is going to take us.
That's a very diplomatic answer. But yeah, I can understand what you're getting at. Okay, well, given that interesting start to the podcast, let's jump straight into the questions. So Kevin, could you give us a quick summary of your background, your education, and your research history, just to let the listeners get to know you a little bit more?
Sure. I mean, I grew up in California, surfing our local beaches. I always wanted to be a marine biologist. And so I went off to the University of California, Santa Barbara, back in 1981, to do my undergraduate. And I've really been here ever since, doing my PhD and then afterwards getting into research full time. I'm an adjunct faculty member here, but then I run a full-time research project for the U.S. government that involves a wide range of topics, and we'll discuss some of the food web stuff today. One of the great things about my job is it's full-time research, and so I spend a lot of time doing research and not necessarily grading exams and things like that. Um, and I get to spend a lot of time in really wonderful field locations. Probably my favorite is this site out in the middle of the Central Pacific called Palmyra Atoll. It's 1,000 miles south of Hawaii. Nobody's lives there except researchers, and we just
Wow.
have nature around us all the time. And so the reason that I love that sort of stuff is I'm really interested in how parasites affect the environment and how the environment affects parasites. Those are the two guiding questions of my research career.
Nice. That's really cool. And I didn't know that you were a one institute guy the whole of your life, starting from bachelor's to PhD to now being a faculty member, all at the University of California, Santa Barbara. That's quite impressive.
Santa Barbara's a lovely place, and we love living here, and I've enjoyed raising my family here, etc. So yeah, come out and visit if you ever get a chance.
I would love to. Thank you for the invite. So now a lot of your work, as you previously mentioned, focuses on mapping trophic dynamics between predators, prey, and parasites. So could you tell us in broad terms what people mean when they talk about trophic dynamics and how you're able to quantify data relating to these studies and put it into your research papers?
So, food webs or trophic dynamics are broadly considered to be ecological maps. They tell us about the flow of energy through ecosystems. They tell us a lot about how communities form and change. They tell us a lot about the effects, for instance, of hunting and fishing, or changes to the environment, or global warming, primarily through their indirect effects. So, in other words, classic ecological concepts like trophic cascades or bottom-up effects, etc. They're all driven through these connections of predator and prey. And so it's not surprising that ecologists over the years have put together many, many dozens of these maps. And they've guided sort of our sort of conceptual framework for how communities work. And so I've been interested in them ever since, as I was an undergrad, and hearing in lecture about classic studies like the way that sea otters help maintain kelp forests by feeding on sea urchins, right? When I started getting into studying parasites, then it dawned on me that almost none of the published food webs included parasites in them. And as a parasitologist, I began to wonder why that was the case. And it was pretty obvious why it was the case, and that is that parasites are small and ecologists aren't trained to think about them. And so they'd really never, it really never crossed their mind to put parasites into food webs.
Hmm.
And that began the start of a whole series of research questions over the last 3 decades that have taken us down into building various food webs with parasites in them.
That's really, yeah, it's very interesting. And I mean, a lot of our listeners will be familiar with the idea of the food chain, for example, but how often does that feature anything parasitic? It very rarely does. So yeah, adding parasitism, which is a very obviously common form of life, into those food dynamics is a very interesting sort of broad direction for your research, and it's definitely one of the things that drew me to it. So, from what I understand, a large part of your work has been focused on the specific role that parasites play in these food webs, as previously mentioned. So, could you briefly explain the perhaps underexplored or underrated impact that parasitism can have on these communities and these food webs?
Yeah, I think that the first place to start is that in any community that we've looked at, there are a lot of parasite species. And so we can start asking questions about, well, firstly, I should say, those parasite species are consumers in the same way that predators are consumers of prey. So we can't necessarily ignore them because they're rare, because they're not rare. They're all over the place. There's probably some people guess that, for every free living species out there, there's another parasite species.
Hmm.
And you know, it's an open question if maybe there's twice as many or half as many, but it's still a lot. So we can't necessarily ignore parasites based on their diversity because they are quite diverse. And if we're going to think about including species in food webs, then by default we might want to include them, unless they're just not important energetically, right? And so that really is the second question, which is if we acknowledge that there are lots of parasites in food webs, then are they important? Are they important in terms of the effects that they have on food webs? And so there was actually a very influential paper came out in 1997 in Tree. Dave Marcogliese and Dave Cohn published it, and it was "Food Webs: A Plea for Parasites" was the title.
Hmm.
And they speculated that there were a lot of reasons to put parasites into food webs. We've mentioned one, which is that they're very diverse. The second is that in some cases we have a pretty good idea that they can control host populations or at least influence host populations. The authors also mentioned that a lot of life cycles are driven by trophic interactions, where the prey host is an intermediate host and the predator host is a final host. And so parasites have clearly evolved to exploit food webs for their transmission. And in some cases, we know that they can manipulate the hosts that are in those food webs, particularly the prey species, through uh, things like altering behavior of prey and so forth. Anyways, they laid out this set of arguments for why we might expect that parasites are important. And we realized that we might be able to answer some of these questions in one of the systems that we'd been studying, a small estuary very close to our campus that had been the field site for many of our PhD dissertations, including mine.
Hmm.
The reason that we focused on an estuary initially is because the diversity of hosts is pretty low. And so we had a good idea of who all the species in the food web were in terms of the predators and the prey. But we'd also been doing a lot of parasitological work, so we had a good idea of who the parasites were also. When we began building that food web, we realized that there was a very appreciable diversity of parasites in those systems. And then we also were able to quantify their abundance in biomass and compare how they compared to the predator and prey in the system. So basically, the upshot of that study, we learned that the parasites in that food web in the estuary changed a lot of the mathematical properties of the food web. And then when you added up their biomass, we realized that there was just as much parasite biomass as there was biomass of the upper trophic levels. So, for instance, the biomass of larval trematode parasites in snails was even higher than the biomass of all the birds that were using that estuary.
Wow.
And so, putting those two things together, it became clear that the biomass of trophic group like parasites gives us a clue that their role from an energetics perspective might be significant. In other words, if we were going to say that the birds are significant players in this food web, then we would have to say also that the parasites are significant players.
Mhm. And I guess that, when you're picturing the upper trophic levels of a food web, the animals generally get bigger. And yet, when you're going up at a level, in this case, in terms of parasitism, they're tiny. So that just shows you that if they're equaling the same weight as the upper level of birds and predators, that there must be so much more of them than perhaps you might have thought to begin with.
Oh yeah, there's there's billions of them really, and the the question that we had was, well, did we just pick a system that has lots of parasites in it, right?
Mhm.
And the honest answer is yes, we did. We picked a
Oh,
system
okay.
that we knew had lots of parasites because that's one of the reasons that we loved working in the estuaries because we could find parasites everywhere we looked. In the many years since then, we've done similar studies in other types of systems, from lakes to coral reefs to kelp forests, and we find the same patterns. It's mostly there are parasites in all of these aquatic systems that we've looked at, and I suspect the same would be true in terrestrial systems as well.
Mhm. Nice. Yeah, it sounds like an interesting system. I think out of the two study areas that you've mentioned before, I think I fancy the sort of Hawaiian island one more than the estuary one, but they both sound very interesting in terms of research. So as we've previously alluded to, when you're talking about trophic dynamics and food webs, there are many famous examples of the importance that predators can have on the ecosystem. You've already mentioned the importance of sea otters on feeding on urchins, sort of increasing the diversity of these kelp forests. One that jumped into my head was also the increase in community diversity that followed a reintroduction of wolf populations into national parks in the United States, and that one I think got a bit famous online. So are you sort of implying or suggesting that parasites can have as much of an impact on their ecosystem and their host populations as predators can in these examples?
Yeah, well, there's a lot to talk about here, and I'll just give three examples. The first
Mhm.
is that um, at least surprising to me, and I think surprising to a lot of people, is one of the things that parasites can provide to food webs is actually food. Um, Many parasites produce these free living stages, and they can be quite nutritious. So, for instance, in our estuaries, fish feed on trematode cercariae, and we can calculate the amount of calories that they get every day from these parasitic stages that are put out into the environment. And many of your listeners will be familiar with chytrid spores and the important role that they play in providing nourishment to zooplankton. So that's one aspect of parasites and food webs that is maybe unexpected to many people. Um, a second one that is, I think, very sexy is when we think about the effect that parasites have on behavior and how that might change food webs. And so, for instance, in those estuaries that we study, the most common parasite effects, or I should say, so for instance, in those estuaries that we study, the most common parasite infects the most common fish
Mhm.
as a secondary intermediate host. And so it lives as a larva on the brain of the fish. And we've shown that infected fish are 10 to 30 times more likely to be eaten by birds than uninfected fish are. And so you could imagine that a parasite is actually making it easier for birds to make a living in that food web by making fish easier for it to catch.
Hmm.
We'd love to do the experiment where we took the parasites out to see if birds did worse, but I think we can at least infer that birds are benefiting and fish are suffering by the fact that you have this parasite that's manipulating the behavior of fish.
That's very cool. Yeah.
A system where we have been able to do experiments is in Japanese streams, where my colleagues have studied the food web there very extensively. And what we see in those streams is that trout are the main top predator, and they mostly eat benthic insects for most of the year. But in October, their stomachs are full of crickets, and the crickets are jumping into the stream, which is not really normal behavior for a cricket. And the trout are feeding on them as they come, the crickets are floating on the surface, and the trout come and feed on them. And this is a very important food source for the trout. It supplies about 60% of their calories for the year. What my colleagues discovered is that the crickets were jumping into the streams because they were infected by an ematomorph worm. This is an aquatic parasite that infects terrestrial insects, and then to get back into the aquatic system, the parasite manipulates the behavior of the host to cause it to go into the water, where it then becomes prey for the trout. And by studying that food web in detail, what my colleagues found was that the parasite changed many different aspects of the ecosystem. Because the trout were feeding on crickets instead of insects, there was more periphyton growing in those streams. And because there was more periphyton growing in those streams, the nutrient cycling was changed. There was also more insects that could break down leaf litter, which changed some of the other processes in that food web. And so the act of a single parasite species, simply from manipulating the behavior of one of its hosts, did some very major restructuring of that food web, and did it in such a way that could be demonstrated with experiments, because my colleagues were able to prevent infected crickets from going into some streams, but allow them to go into others. And so that, I think, is probably the best demonstration of an effect of parasitism at the food web level with a real field experiment.
Wow, that's really, I mean, 60% of the calories for the year is staggering, especially considering it only happens in the autumn time, as you mentioned. Um, yeah, I guess that, there is a bit of knowledge, I guess, in the public consciousness about sort of brain controlling fungi, especially with recent TV. I'm not sure if you've watched The Last of Us, but that's a series that has been quite popular, and that's about, fungus controlling people's brains. But this is an example that is so much more useful in studying ecosystem dynamics, and it's happening and having a massive effect on these both terrestrial and aquatic ecosystems, I can imagine.
It's funny, if you did try to make a TV drama based on that life cycle, nobody would believe it. It would just be
Yeah,
too far-fetched.
yeah.
So yeah.
I mean, it
Uh,
is baffling. Yeah.
yeah. I will say that, it's certainly one of the motivating factors behind working on parasites is that um, they're just so creepy and weird that a
Mm.
certain personality becomes attracted to them, and I've always found them entertaining and fascinating in that way.
Well, I think we share that personality defect, let's call it. So moving on from this sort of behavioral aspect of parasitism in communities, one area of your research that also interested me was the studies into the life cycle and ecological dynamics of the parasite schistosomiasis. Now, this might be a lesser known parasite to the general public, but it has a terrible impact on millions of people every year in tropical regions. Could you explain a little bit more to the listeners about this parasite and its unique life cycle?
Yeah, I think that as with any parasite ecological study, the life cycle is key, right? So
Hmm.
understanding the life cycle is the first thing you need to know if you want to intervene in a way to benefit public health for a disease like schistosomiasis. And so schistosomiasis is caused by three different species of trematode worm in the genus Schistosoma. They all have a pretty similar life cycle where they use a snail as a first intermediate host. Unlike a lot of trematodes, they do not have a second intermediate host,
Hmm.
They are able to transmit directly from a snail to the final vertebrate host through the larval cercarial stage, which then penetrates, well, the ones that we're concerned about penetrate humans. Um, the larva then migrates into the circulatory system, and the adults pair up their separate sexes, and they basically live in permanent copulation inside the circulatory system, and they can be quite long-lived. They can live for a dozen years or more, as far as we
And
know.
how do they penetrate the human body when they're transmitting from the aquatic environment into the human circulatory system?
Right. So the cercarial stage is produced inside of the snail. It's released every day, and they have pretty good sensory capabilities. They're able to detect light and water motion and things like that, and also chemosensory abilities. So they 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 find the skin of a human that's working in the water or bathing or swimming or whatever. And then they have enzymes that allow that cercaria to start penetrating into through the skin and eventually end up in the circulatory system. So basically, you've got these worms. They live in your circulatory system for a dozen years, and they don't really take a lot from you. I mean, they eat red blood cells, but they don't cause, in most cases, a lot of anemia or anything like that. Their impacts on human health have to do with the eggs that they produce. And the reason that they cause pathology is the eggs have to get from the circulatory system out into the environment. And that's not an easy thing to do. There's no easy way out, so to speak.
Hmm.
And so they have a fairly, I would just say, unsophisticated or coarse mechanism where they they the eggs circulate, and for one of the species, the one that causes the most damage, Schistosoma manson, they end up in the renal system, the portal veins around the liver, and then they are able to penetrate through the small capillaries using a hook that sort of causes damage in the circulatory system. That creates a little wound, which leads to blood that can leak out. But it also stimulates the host immune system. And so the host responds by trying to lay down a bunch of immune response, which is often a calcified response, layers and layers of host cells and calcium that coat these eggs. And so many of these eggs end up in the liver, and the host immune system attacks them, and they fail. Most of them fail to get out of the host. But the host immune system, to solve a small problem, a tiny egg, creates a much bigger problem. It creates a calcium nodule that's many times the volume of that egg. And over the course of many years, with literally, hundreds of eggs per day per worm pair coming out, you can get millions of eggs filling up and destroying the liver function. That's just one of many effects that this disease has, but it's a chronic disease that has long-term impacts for people that are chronically infected.
Hmm.
And people have done a lot of studies that have shown that it can impair learning in children. It stunts their growth, leads to malnourishment. It can lead to early death. It can create one of the species, Schistosoma hematobium, promotes vaginal and bladder cancer due to all the inflammation that it creates. We could go on with a long list of pathologies. And the distribution of schistosomiasis is pretty localized to tropical areas of Africa, Asia, and a little bit in Latin America. It mostly affects people who are in contact with the water and don't have good sewage treatment access. And so because, obviously you have to complete that life cycle, humans need to defecate or urinate into the water where the snails live, and that is a condition of basically poverty. And
Hmm.
so Schistosomiasis is one of the main neglected tropical diseases in the sense that our sort of modern medical enterprise doesn't think about it very much in Western nations because we're really not exposed to it. So the reason that this moves into a food web study is I'll get to that in a second, but I wanted to say that one of the great developments in schistosomiasis public health has been the discovery of a medicine called praziquantel, which is an anti-trematode drug. It's very effective. The drug companies make it available for very little money.
Hmm.
And so you can go into a village and for $1 or $2 per child, you can cure everybody in a very simple way. And
Hmm.
so you would think that that would solve the problem. And unfortunately, it, taking that medicine cures you of the adult worms. It doesn't do anything about the buildup of eggs and pathology that you have in your body, and doesn't prevent you from becoming reinfected the next day.
Hmm.
And so you can imagine you're a kid living in sub-Saharan Africa, and what are you going to do after school? You're going to go play in the river like all your friends do, and you become reinfected the next day after your treatment. So mass drug administration, although it has had incredible benefits, has not necessarily been a way to eliminate schistosomiasis. To do that, basically, you would need to do a lot of investment in public infrastructure and hygiene and et
Hmm.
cetera. The thing that has worked very well in many places over many years is snail control. So there are many ways that people can impact snail populations, and many ways that people can benefit snails indirectly. So, for instance, um, if you are at a place and you're putting a lot of sewage into your lake or your pond, that's really great for snails. They love eutrophication, and so they grow. But you can also poison snails with certain sorts of chemicals, so you can get rid of those snails in your pond through those applications. And we've done research that have shown that by far the most consistent and most effective way to get rid of schistosomiasis is to deal with the snails.
Hmm.
You want to combine that obviously with drugs because you want people to be healthy, and
Hmm.
um, but in terms of dealing with like the overall infection risk at a particular place, snail control is really the most effective thing. Okay.
Yeah. I guess that another comparison for this sort of vector control approach would be uh, managing mosquito populations to reduce malaria. Now, you can obviously treat malaria in the people who are sick, but this is what I think a really interesting example of where ecology comes in to real clinical problems that are sort of blighting the world, that we can integrate the ecology and the understanding of the vectors, the snails and the mosquitoes, into the treatment of this horrible under underfunded disease, I guess.
Yeah, I think that's a great comparison, and there are others with
Mhm.
tick transmitted diseases and various other types of vectors where ecologists have a role to play, working alongside medical professionals. The medical professionals are treating people, which is really important, and the ecologists can think about treating those life cycles
Mhm.
and thinking about potential interventions. So. We've been interested in schistosomiasis for a long time, and one of the most famous cases of a schistosomiasis epidemic has been along the Senegal River in Senegal. And the history is one that's been repeated in many places, which is that a World Bank funded project came in to build a dam across the Senegal River with the idea that they could block the tide from coming up the Senegal River and create a lot of new arable land for agriculture, primarily sugarcane plantations. So the dam went in. The area upstream of San Luis became more arable for sugarcane plantations. Some of the landowners, you know, had a great new business there, and they brought in all sorts of laborers, mostly from the south of the country. Now, those laborers, when they came in, they were from places where schistosomiasis was endemic, and many of them were infected. And so they brought schistosome worms with them, and they introduced Schistosoma mansoni into the Senegal River. In addition, the dam created lots of great habitat for snails because it was now more fresh water. There was less tidal influence on it, and lots of aquatic vegetation that the snails love, etc. And so that created the largest epidemic of schistosomiasis that the world has ever known. Started in the mid 80s, and it's still going on today.
Wow.
So obviously, there's a lot of investment in treating people, and we were interested in understanding a little bit about the ecology. In particular, is there something about the food web that's changed to benefit snails? And so I've mentioned, better snail habitat, also probably more eutrophication from people living along the river,
Hmm.
uh, and fertilizers coming in from sugarcane and et cetera. Um, but one of the things that we proposed was that the dam had simplified the food web in an important way, and that is that before the dam went in, there used to be a fishery for a giant river prawn, Macrobrachium vollenhoveni. And it turns out that prawns and a lot of crustaceans, for that matter, love to eat snails. And so we wanted to see whether or not this genus of prawn was an effective predator on snails. So we did a bunch of laboratory experiments, and what we found was that if you put snails in an aquarium, they do just fine, but you throw a prawn in there with them, and the prawn eats all the snails.
Hmm.
Okay. And that was, it's an aquarium study, so who knows what happens out in nature. But we wanted to see whether or not we could potentially use the native river prawn as a control for snails in Senegal.
Hmm.
So we did an experiment where we went to two villages in Senegal. And the first thing that we did was we asked if we could treat all the children in both villages for schistosomiasis. And so we got the kids, we checked their urine and feces for parasite eggs, quantified how infected the kids were. We treated them all so that they were all then uninfected, and then we let them go back to school. We then, at one of the villages, we put out an enclosure at the area where everybody goes to swim and do the laundry and get water for the animals and bathe and stuff. And in that enclosure, we added giant river prawns. And then we tracked the abundance of snails at both villages over time. And not surprisingly, where we put the prawns, there was a much lower abundance of snails than in the place where the prawns were not put.
Hmm.
And in particular, the prawns really like to eat infected snails. So there was a much lower density of infected snails at those places where the prawns were present. I think primarily because infected snails just don't respond to predators as well.
Is that a similar sort of effect to the previously mentioned fish that were infected, becoming more predated by the birds?
No, it's different in the sense that the prawn is not a final host, so it's not in the parasite's interest for the prawn to eat it. It's actually quite different. I'm glad you asked that
Hmm.
question. We had seen this in the aquarium studies as well, that the infected snails are they- they're pretty, I guess you could just imagine they're sort of sick in a way
Hmm.
that.
Yeah, just lowered fitness, lowered ability to evade predators, generic stuff like that, I guess.
But the most important part of the experiment is we could come back later and test all those kids. So we brought the kids back in, we had them provide samples, and then we treated them again. But then comparing the samples before and after treatment in the two villages, what we found was that um, the initially, the village that had the most parasites was the one where we put the prawns. That
Hmm.
was for ethical reasons. We figured that's where it could potentially do the most good.
Hmm.
And those kids were then much less infected than the village where we didn't put prawns,
Hmm.
even though the village where we didn't put prawns started out as less infected in the first place.
Okay.
So basically, the prawns had a direct effect on reducing snail populations, and that had then an indirect effect on preventing or at least reducing reinfection in the school kids at those sites. So this was just, we thought, an interesting example of how the dam um, affected the food web. And I didn't actually mention how the dam infected the prawns. That's an important part, which is that these prawns in the Macrobrachium genus, they breed in estuaries, and then the larvae walk upriver and live in the freshwater systems.
I see.
What the dam did was prevent the migration of the prawns from coming up into the areas where the people lived. And
Hmm.
eventually, the prawn population became very diminished. The fishery for the prawn went away, and people forgot all about the prawns. They didn't. The younger fishermen had never even seen one.
Wow.
So that then led to the question: well, what can we do? Can we reintroduce the prawns? Obviously, as far as the if the dam is still there, the prawns will never be able to migrate back down. And so we decided to see if we could figure out how to do aquaculture for the prawns because allowing the villages to raise prawns at their village could help them get some income and also help reduce schistosomiasis. We spent 3 years trying to figure out how to do that, and it did not prove to be easy. It was
Yeah.
much harder than expected.
Hmm.
So that's unfortunate. Um, and one of the reasons that this was difficult is because the prawns really needed better water quality than was available for them at these sites. So it turned out that at these sites where people get infected, there's a lot of eutrophication, there's pollution from detergents, there's all sorts of stuff that makes it difficult for prawns to persist there.
Hmm.
The other approach that we looked into, which was sort of my favorite, is could we build a bypass for the prawns on the dam?
Like a salmon elevator. I think I've seen one of these. Yeah.
And we do this for fish. They build them for eels. They build them for here in the West. They build them for salmon. And so I actually went out to Senegal with an engineer who had designed eel ladders, and we came up with a prototype that we think would have worked pretty well for prawns. We were not able to sell that idea to funders,
Hmm.
unfortunately, to actually put it into practice. Um, at this point, my main concern is that the places where people are being infected are not really great habitat for prawns, and so it's not clear that
Hmm.
without doing additional work at these sites to sort of improve water quality, that prawns would be as effective at reducing schistosomiasis as we had hoped they would
Hmm.
be. We have studied other possibilities like removing some of the aquatic vegetation that the snails really love, and that seems to be pretty promising, actually. So there
Nice.
are other ways to intervene in a food web.
Hmm.
We were hoping to exploit the top-down approach, but it
Hmm.
turns out that the bottom-up approach is also an option that might even be more effective.
I guess that's the sort of the best part of these sort of super complex food webs that there's so many interactions that if one of the targets that you sort of fund or research doesn't work, there are alternatives. I just find this case study so interesting because it's such an interesting intersection of ecology, economics, development of underdeveloped nations, and it must be really gratifying to work in such an interesting system that can have such a profound impact on people's lives. I obviously enjoy working on my study species, but to have such applications as yours, yeah, I can imagine it can be quite fulfilling.
Well, I think we learn the general ecological lessons in places like a salt marsh,
Hmm.
where there's no real human health issues.
Hmm.
And hopefully we take some of those lessons and we can apply them to other cases that have more practical,
Yeah,
particularly human health outcomes.
lovely. Yeah, I think that's a great note to finish on. But just before we go, I always like to finish these podcasts with a slightly, more light-hearted question. So you talked earlier about the sort of using parasites as a source of food. Um, Now, parasites do constitute a significant part of some species' diets, but do you think that a parasitic species could ever become a viable source of food in human diets, for example?
I mean, firstly, I think we just need to realize that we are all eating parasites all the time.
Yeah.
Certainly, people that are carnivores and eat,
Mm,
fish and meat are exposed to lots and lots of parasites. In fact, um, probably one of the main reasons that people developed fire as a technology, in addition to
hmm
keeping warm, was to help them kill the parasites in the food they were eating. I mean, that wasn't necessarily their intention, but
hmm
it certainly made food a lot safer to eat
And
because
tastier.
before that, we
Yeah.
were getting exposed to
Yeah.
tons and tons of stuff.
Yeah.
Um, and so if you're like me and you like to eat sashimi,
Mm,
you're really um, putting yourself more on the front line in terms of eating parasites. And of course, that's a real risk. Although these days, most of the sashimi that you'll eat anywhere around the world has been frozen and those parasites have been killed. So eating sashimi is a lot less exciting than it used to be
yeah.
from a
That's one
human
way of
risk
putting it.
standpoint.
Yeah.
Yeah. And we in the lab we will sometimes initiating a new graduate student or something like that will you know see if we can eat a particular parasite. We always make sure that we cook them first, and I'll just say that anything is actually pretty tasty with enough butter and garlic, so
I was
that
gonna say, yeah.
go ahead and try this at home as far as I'm concerned.
Well, this is making me very glad that I'm not a new initiate at your lab, but it sounds quite an interesting sort of gastronomical experiment at least.
There's actually there's a liver fluke in deer
Hmm.
that is commonly eaten intentionally by deer hunters.
Oh, really? Okay, I didn't even
Yeah,
know
it's
that.
it's a big it's a big fluke, about the size of your thumb. They call them butterflies,
Okay.
and yeah, and they eat them. So parasites are actually parts of cuisine, even in modern day.
I guess that's the question answered then. It's already in the human diet and we just maybe don't even know it. Okay, so thank you for a really interesting talk, Kevin. I really appreciated it and found it so interesting and I think very eye-opening at times. So thank you so much for coming on.
Well, Joe, it was my pleasure. Keep up the good work. I love what you're doing, and it's. I will say that podcasts are a a really nice way to be able to share research in a more casual way. I'm delighted to be invited to share mine with you and your listeners, and have a great day. And hopefully, we'll get to meet in person sometime.
I would love that. Thank you. That's very nice of you to say.
All right, bye.
The ParAqua podcast was funded by COST, the European Cooperation in Science and Technology, and is a product of the ParAqua Action Group. If you are interested in joining an action group or just want to know more, please go to www.paraqua-cost.eu or follow the link in the episode description. The podcast is produced by myself and Serena Rasconi, and the Science Communication Coordinator is Anna Gavrilovich. Once again, thanks for listening, and see you next time on the ParAqua Podcast.